All right, now let's continue our discussion of chapter 8, chapter on photosynthesis in our history lesson here. So we looked at Jan Ingenhausen, who noticed that plants give off these bubbles of gas only when they're exposed to the light. Thought that was a curious result. Still in the 1800s, Julius Mayer, he hypothesized that plants essentially turn sunlight energy into chemical energy, light energy into the chemical energy in carbohydrates. Um, Again, this was just a hypothesis. He really didn't have the abilities or know how to test this at that time, but his hypothesis, as we'll see, turned out to be correct. Um, Melvin Calvin. So now we've jumped up quite a bit to the mid-1900s, and his name has become identified with what we know as the Calvin cycle, which is the process by which plants take in CO2 and use energy they have derived from the sun to make sugars. So his work essentially in part supported the original ideas of Mayer that plants convert chemical energy or sunlight energy into chemical energy, and specifically the carbon that ends up in the sugars comes from carbon that's in CO2 in the air around us. Rudolf Marcus, 1900s, 1990s to be more precise, figured out the workings of the electron transport system, which we'll talk about in a minute. This is the process by which plants take that sunlight energy and using pigments in the thylakoid membrane, this particular part of the chloroplast, they essentially do that initial conversion of sunlight energy into chemical energy, in particular the movement of electrons from one molecule to another. Iwata and Barber. So we of course know that plants need water, and they figured out the process by which plants split water, doing what's called photolysis, the splitting of water using photons of light, and essentially figured out that the oxygen that is released by plants, that oxygen comes from water. So essentially the water that's in the ground, the plants split that molecule up and spit out the oxygen as O2 gas, what we need to breathe, of course. All right, let's move on to section 2, more of the details of photosynthesis. So essentially, a lot of biochemists, bioengineers are trying to figure out how to build systems that can take sunlight energy, convert it into a form of energy that can be stored, essentially mimicking what plants do, mimicking photosynthesis. So with plants, it kind of starts with chlorophyll, this pigment, this molecule that absorbs sunlight energy. And when it absorbs the sunlight energy, these energized electrons are released from the molecule, specifically this region right here. There's this magnesium in the center here. And when the sunlight energy strikes this part of the molecule, the magnesium begins to vibrate faster and faster and gets so excited that it essentially loses electrons. So this is sort of the reactive part of the molecule. Now over here you see this long chain of carbons and hydrogens. This is the non-reactive part, but this is the part that sort of anchors the molecule in the inner membrane of the chloroplast. So chlorophyll is the main photosynthetic pigment, but there are several kinds. There's chlorophyll A and chlorophyll B, and then there are other types of pigments in plants. There's these things called carotenoids, for example. And here we have different wavelengths of visible light. Okay, that part of the electromagnetic spectrum that we can see, and that are the parts of the spectrum that plants make use of. But you can see within the visible spectrum, not all wavelengths are created equal. What we're seeing here is what's called the absorption spectrum. So where you have a peak in the line, like right here or here or here, that's where that particular pigment is absorbing a lot of that particular wavelength. And down here in the middle, around the greens and yellows, you can see the lines are very low. So in those portions of the spectrum, the plants are not absorbing those wavelengths. So plants absorb at the red end and the blue end, but not in the middle. So what's happening to these wavelengths of light? Well, the plant is essentially reflecting them and also transmitting some of them. That is, some of those wavelengths are just bouncing off the plant and some are just going straight through. This is why plants are green, because they are reflecting a lot of the green wavelengths and gives them that green color. Now, the action spectrum, this figure is not in your book, but I wanted to mention it. Again, we've got our different wavelengths, and this shows the portions of the visible wavelength, the visible spectrum of light that provide the most energy for photosynthesis, that is, cause the most action on the part of that process. So when plants are exposed to blue wavelengths, that provides a lot of energy. Red wavelengths, that provides a lot of energy. But again, the greens, not so much. I mean, there's some photosynthesis going on, but not nearly as much as the other wavelengths. So, plant structure. Of course, you know that plants have stems and leaves. And when it comes to photosynthesis, for the most part, leaves are where the action is. It's a plant structure, a plant organ, if you will, whose job is to do photosynthesis. It has lots of surface area to absorb that sunlight. And although they might not look like it, of course, plants have a three-dimensional structure. They have depth to them. And there's all these cells inside them, these mesophyll cells, whose job is to do photosynthesis. These mesophyll cells are loaded with chloroplasts, these green dots here that have lots of chlorophyll inside them. The chloroplast, as we'll see, has a complex membrane inside of it, known as the thylakoid membrane, provides lots of surface area to absorb sunlight. The thylakoid is where the chlorophyll is, and this is just a picture from an electron microscope of a chloroplast. Let's look a little closer with the chloroplast. So notice the chloroplast has two an outer and an inner membrane, so there's two membranes covering it, so thus a chloroplast is a membrane-bound organelle. And the liquid inside the chloroplast is known as the stroma. And then here's the thylakoid. The thylakoid is the membrane on the very inside of the chloroplast. It has a structure think of it as it's arranged in these flattened discs that are sort of stacked up. So the whole thing is known as the thylakoid membrane. And the stacks of these flattened discs are known as a granum. And you can see there's connections between the different granum. The space inside of these discs, essentially inside of the thylakoid membrane, is known as the lumen. And we'll see that the stroma and the lumen, there's different things happening in these different parts of the chloroplast when we get to some of the details of the process. All right, so we already talked about the work that this guy did, this electron transport chain. We'll see that essentially electrons, when they're released from the chlorophyll, they don't just sort of go out into space and hang out. There's something there to grab onto them, these energized electrons. And what's going to happen is these energized electrons are going to be transferred to these electron carriers, and we'll see in photosynthesis, NADPH, this molecule, is the electron carrier, is the primary electron carrier. And you don't have to memorize what these look like, but I just wanted you to see that they are, of course, organic molecules, lots of carbon and hydrogen, some nitrogen, and some phosphorus as well. And NADP, so this is the form that's sort of lacking an electron, and you can see it has a positive charge. And so what happens is the NADP gets converted into NADPH when it essentially has an electron. Whoops, I don't want to have it as a plus, it's a minus. When it has an electron added to it, it also gains a hydrogen ion. So notice up here. So it's gained an additional electron and it's gained a hydrogen ion, and it's converted from NADP to NADPH. It's going to carry an electron. And when we look at sort of the overview of photosynthesis, you see here, there's our NADPH. It's going to carry electrons from the thylakoid membrane to this process that's occurring in the stroma, the liquid that surrounds the thylakoid membrane, what are known as the light-dependent reactions. So photosynthesis can be broken up into the light-dependent reactions and the light-independent reactions. On the thylakoid membrane, we have the reactions that are occurring that depend on light. And over in the stroma, we have the reactions that do not depend on light. Okay, but as you can see, they make use of molecules that are produced in the light-dependent reactions, these energized molecules. And so what the light-independent reactions are going to do is use energy from NADPH and ATP to take carbon dioxide out of the air and build sugars, and in the process send these lower energy molecules. So now ADP and NADP plus have less energy than NADPH and ATP, and they go back to the light reactions, to the thylakoid, to be re-energized. And so they're just traveling back and forth in their lower energy state, higher energy state, lower energy state, just back and forth from the light reactions to the light independent reactions. Notice over here in the light reactions, that's where photolysis occurs, where water is split and you create some oxygen in the process. We'll look at the details of these two processes in our next video. I'll go ahead and stop here. We'll look at the details of how water is split, how the NADPH and the ATP are produced, and how they're used in the light-independent reactions that here, you may recall, this is what's known as the Calvin cycle. Melvin Calvin was the one, he and his co-workers figured out how the CO2 is essentially incorporated to make sugars. Okay.